Sedimentary rocks are the richest source of fossils, and the relative sequence of fossils in sedimentary rock, or the strata, tells us the order in which fossils were formed, although it doesn't really tell us their ages. And the fossil records show that there have been great changes in the organisms that have lived on Earth. Many past organisms were unlike living ones, and ones that were once common are now extinct, and new groups emerge. You get the general idea. So while the fossil record is not complete, it does provide a detailed account of biological change over geologic time, and we can see that sort of progression. But remember that the cell theory says that all cells came from pre-existing cells. Well, where did the first ones come from? Well, there's a couple of different major theories to that, but the most supported one, the one we have the best evidence for, is what's called abiotic biosynthesis. So the base idea for this is quite old. Early organic chemists proposed sort of this sort of idea, and in the 1920s, two Russian chemists, Operin and a Russian and a British chemist, Operin and Haldane, came up with this idea. They independently postulated that conditions on early Earth favored the synthesis of organic compounds from inorganic precursors. Basically, the atmosphere was really low in oxygen. It was a reducing atmosphere instead of an oxidizing atmosphere. Um, so that's going to make a difference. Young suns emit way more ultraviolet radiation, so the lack of an ozone layer would have allowed more of that radiation to reach the Earth. And then the great deal of sort of water flow and dissolved materials would have made the oceans and bodies of water very high in nutrients, creating a perfect environment for cells to form. Years later in the 50s, Miller and Urey tested this idea by creating in the lab conditions that had been postulated in early Earth. They discharged sparks as atmosphere of gases and water vapor, and so we can see that sort of basic idea. So this is really looking at that primordial soup, giving it heat and electricity, and then looking at what happens over time. And when they actually extracted that material, they found a large number of things like amino acids form spontaneously within that structure. Okay, so basically, laboratory simulations of early Earth conditions have produced organic polymers. Now, we did go back and we realized eventually that the gas mix that they used wasn't quite right. We had that idea wrong. They redid it years later, and they still got similar results, not quite as good, but we get the general idea. They also went back and retested in the 2000s the samples that Miller and Urey had extracted, and we found that they were much more successful than they originally thought. And that could be that the machines are just more sensitive now, so we're able to pick them up, or it could be that just over time more continued to accumulate even without the addition of electricity. So take that how you want. Okay. But once you have those basic macromolecules, they can then come together to form protocellular structure. So basically, life is defined by two properties, reproduction and energy processing. Life can't persist without both of them. Okay, so early sort of assemblies of these vesicles and structures can form spontaneously when things like lipids or other organic molecules are added to water. The hydrophobic molecules form a bilayer, basically a droplet surface, that could be aided by other non-organic things like the montmillerite clay we can see here on the example would vastly increase the self-assembly production, but you're going to end up with some sort of container. That's our first real step. We have macromolecules, now I have a container. I have sort of the base cellular structure. Protocell, early cell. Coacervates are one type of early cell, and that's a droplet that forms spontaneously when you have enzymes sort of trapped. Coacervates, they can get increasingly complex. They're able to sweep up chemicals from their surrounding environment. They're able to bubble off. So we're seeing some low level of reproduction and energy processing. They can get more and more complex over time, but abiotically produced vesicles are going to be those next major steps. Okay, once I have actual containers, my next major step is going to be some sort of genetic material. Okay, the genetic material is the first genetic material is proposed to be RNA. So uh, RNA is a catalyst, so it's able to catalyze its own reaction like an enzyme. So RNA can act like a ribozyme, is the word here. It's a RNA that is enzymatic. It is simpler in structure. It's single-stranded typically. So basically, once we have RNA, all the properties that are needed for a molecule that could catalyze and produce early life, RNA meets those components. Although RNA seems well suited to form the basis for self-replicating chemical catalysts, it's unlikely that it was the only kind of molecule to do so. From a purely chemical one, it doesn't really make sense. But there are other polymers that resemble RNA but that are chemically simpler. We don't have any remnants of these compounds in present-day cells, and something like that wouldn't leave a fossil, but it's likely that there were precursor steps. And then we see that sort of transition over time. The most successful protocells would be the ones that had these RNA structures, because then they would be able to carry genetic information and they'd be able to vastly speed up this feedback loop. Once RNA sequences that carry genetic information appeared, further changes are possible. Like that RNA could then be refined over time, and eventually you could switch from an RNA world to a DNA world, but those steps are possible. There are other theories, but these are the most supported hypotheses. Once we had genetic material, that really allowed for much more complex structures. Okay, so once we had that genetic material, that then would move us into the sort of heterotrophic idea. The heterotrophic hypothesis is the proposal that the first living organisms were heterotrophs. They can obtain their energy by feeding on others. There are likely to be sort of, before there were other organisms, they would feed on surrounding leftovers of their origin, things like fermentation and that kind of thing, but then cells would begin to eat other cells. We know heterotrophs came first because we know when oxygen evolved, when photosynthesis evolved, we can see that difference. So the oxygen revolution that we can see in the picture on the right, that is going to be when autotrophic organisms evolved. You can see it was a very quick shift, because it's a highly efficient energy form. A good sign of that is that, well, before this point, we don't see rust in our rock. And then after this point, hey, look, rust in rocks. So we know when oxygen evolved. So the heterotrophs definitely came before the things like cyanobacteria, the early autotrophs. Those are the actual ancestral organisms of the mitochondria and the chloroplasts. The mitochondria and the chloroplasts have similar evolutionary origin, and this is where we get to the idea of the endosymbiotic theory. The endosymbiotic theory states that an early ancestor of eukaryotic cells engulfed an oxygen-using non-photosynthetic prokaryote. That engulfed cell became dependent upon the host. So basically it engulfed it and probably digested, engulfed and digest, but eventually one wasn't digested and it began to produce ATP. It became a beneficial relationship, and that organism had more energy, could survive and reproduce, and dominated the ecosystem. Okay, plants went on to do that again with photosynthetic organelles, the chloroplasts, and that is going to be where that came from. How do we know this? Well, if you actually look at a mitochondria and a chloroplast, the inner membrane is prokaryotic, the outer membrane is eukaryotic. If you look at how they divide through binary fission, not meiosis-mitosis. So they divide like a bacteria. They have bacterial-sized ribosomes. They have bacterial circular chromosomes. So the mitochondria and a chloroplast are good examples of endosymbiotic organelles, because they were a prokaryote and they became part of a eukaryote, an endosymbiont. Okay, we get the general idea there. So that endosymbiosis is a very foundational theory. The eukaryotic cells differ in many aspects, but these really allowed for protists and fungi and plants and animals to evolve because they gave the energy for complex life. Okay, animal cells really only did endosymbiosis the one time. Plant cells went on to do it multiple times, and that's why they have more diverse organelles within their structure. They actually have a whole group of organelles that came in through endosymbiosis called the plastids. Okay, and the chloroplast is just one type of that. Basically, you have the proplastid, which is like a stem cell, and it can become the different types of plastid. Chloroplast, photosynthesis, chromoplast, pigment, amyloplast, starch, amylose, aleoplast, lipids, proteoplast, proteins. You get the general idea. So all of those plastid organelles came about through this endosymbiotic process. They didn't just do it they did it lots of different times. And that's one of the major regions that plant like protists and plants and fungi are so different from each other because they went through different levels of endosymbiosis, different secondary endosymbioses. Everything went through primary endosymbiosis. That's the one that did the first one that actually came like us with the mitochondrial structure, but plants have secondary and we don't. Okay, once you have the energy capability of these endosymbiotic structures, that's where multicellular life is possible. So once you have one cell with enough energy, it can't get bigger and bigger and bigger, so it divides. Okay, experiments have shown that groups of microbes secrete useful molecules that all members of the group can benefit from. So groups of macromolecules are going to grow faster than ones that are not grouped. Okay, freeloaders are going to be problematic, so we're going to see some of that evolutionary pressure there. But there's many different hypotheses for how multicellular life evolved. Another hypothesis is that groups of function-specific cells aggregate into masses that then form a multicellular unit. That's basically what slime molds do. Another hypothesis is that basically primitive cells underwent nuclear division without actually dividing the cell at first, which created sort of different sort of subsets of cellular space. You don't really need to know these theories, but how did multicellular life evolve? Lots of different theories exist here, and the problem is that when you look at all the different branches of life, many different types of life are multicellular, and they did not evolve that structure from a common ancestor. This is not a last universal common ancestor thing. Multicellularity is so energy efficient and so advantageous that it evolved separately several times. So each of these red sections are ones that have evolved it. And you notice there's no central structure. Okay, so it's entirely possible that many of those theories of multicellularity are true. But a great range of eukaryotic cells evolved and formed the diversity of life that we see now. Comparisons of DNA sequences suggest that there is a common ancestor, LUCA, the last universal common ancestor that evolved about 1.5 billion years ago. But because the first multicellular organisms were simple, soft organisms lacking bone or shell or other fragments, they're not really well preserved in the fossil record. So it's going to be very hard to find definitive proof. But the theory and the evidence that is there is strongly supported.